Hello, everyone. Thank you all to join my talk about solutions to facilitate the development of CAR-NK cell therapy. I am Tong Ho. I'm an associate product manager at Sino-Biological, and I'm in charge of product development and product line optimization. In this symposium, I'm so happy to share with you some updates from us regarding the CAR-NK cell therapy. I will break down my talk into four parts, which begins with a brief introduction of CAR-NK cell therapy and a comparison between CAR-NK and CAR-T. In the second half, I will emphasize some key elements in the CAR-NK production and what we could do to facilitate the production. So let's go with the first part, CAR-NK cell therapy. First of all, speaking of CAR-NK cell therapy, we have to know why NK cells are so special and important to be an approach of cell therapy. Since I think all of us have got understanding on NK cells, we'll just take a very quick look at the importance of NK cells here again. As you can see here, as the largest lymphocyte population representing innate immunity, NK cell, which is natural killer cell, perform diverse functions as follows. NK cell can interact with dendritic cell and T cells by the interferon gamma secreted by NK cell. NK cell can also kill virus-infected cells or bacteria-infected cells. In addition, NK cell can also kill bacteria directly by granzyme and porphyrin. By using the innate cytotoxicity of NK cell, CAR-NK has also become a new approach and provide new opportunities for cell-based therapies. The NK cells need to be activated before they can kill tumor cells or other infected cells. So here we can see there are two major ways NK cell can get activated. First, NK cell can bind to target cell through both activating receptors and the inhibitory receptors. The activating receptors could induce the activation and the cytotoxicity, while the inhibitory receptor would silence NK cells. The status is dependent on which signaling is dominant. Therefore, the overall status of NK cell is determined by the competition between the activating and the inhibitory receptors. In addition, NK cells also express CD16A on the surface to bind the target cell-bound antibodies, which can activate the cytotoxic activity of NK cell. This is a process known as ADCC. Once the NK cells get activated, they have the ability to kill target cells. Activated NK cells mainly kill target cells through three mechanisms. The first is through the release of cytotoxic particles such as porphyrins and granzymes, which induce the target cell lysis or apoptosis. The second is through the expression of Fas ligand and TNF-related apoptosis-inducing ligand. So once the NK cell gets activated, it can induce the apoptosis of the cells which express Fas or TRIL receptor through a cascade reaction of endogenous enzymes. Moreover, activated NK cells can also secrete cytokines to amplify the immune response. These cytokines can bind to other immune cells, which can lead to their activation and proliferation. As we know, the NK cells are very potent to induce cytotoxicity and kill tumor cells. CAR-NK is now under consideration to further facilitate the potency of NK cells. By adding the CAR molecule, NK cells can target the cancer cell better and specifically. Once the NK cells are directed to cancer cells, NK cell will be activated and perform cytotoxic activity by all the other related receptors, such as activating receptors or FASL, and ADCC effects. The design of CAR molecule is almost the same as it is used in CAR T cell, which includes four parts. Extracellular antigen binding domain. This domain is usually derived from an antibody that binds specifically to a particular antigen expressed on the surface of cancer cell. The antigen binding domain is responsible for recognizing and binding to the cancer cell. The second part is the hinge region. The hinge region connects the extracellular antigen binding domain to the transmembrane domain. At the same time, provides flexibility to the CAR molecule to make it move freely. The third part is the transmembrane domain. This domain is a hydrophobic region that anchors the CAR molecule to the membrane. The fourth part is the intracellular signaling domain. This domain is typically derived from signaling proteins that can activate the immune cell upon recognition of the cancer cell. So after binding to the antigen, this intracellular signaling domain will initiate a signaling cascade that can activate the immune cell to kill this cancer cell. So next, let's take a look at the comparison between CAR-NK and CAR-T. Unlike the well-established CAR-T production, CAR-NK is still a new approach and needs a lot of optimization. However, theoretically, CAR-NK has a lot of advantages over CAR-T, which makes it a promising alternative cell therapy. The first and important thing is CAR-NK would have a lower cost. It is because of the cell source of the CAR-NK cell. Because the starting cell source for CAR-NK cell therapy is natural killer cells, which are present in peripheral blood and can be readily isolated and expanded. In contrast, the starting cell source for CAR T cell therapy is T cells, which are much less abundant in peripheral blood and require more extensive processing to isolate and expand. The second thing is CAR-NK has a good off-the-shelf availability because NK cells can be obtained from a variety of sources, including cord blood, peripheral blood, and cell lines, which allows for the development of the off-the-shelf CAR-NK cell products. And what's more important, it also does not need to obtain the NK cell from the patient. This eliminates the need for individual cell manufacturing and reduce the overall cost of production. Another advantage is CAR-NK cells are safer. Since conventional T cells require HLA matching to recognize the target cell, so GVHD, which is graft versus host disease, is one of the prevalent toxicities of CAR T cell therapy. Since CAR-NK cells act independently of the HLA matching to recognize the target cell, GVHD has rarely been reported in clinical or preclinical studies of CAR-modified NK therapy. CAR-NK cell therapy also have other advantages, like it will have lower risk to cause CRS or neural toxicities, and it also has higher efficacy on solid tumor. So now let's go to the key elements in CAR-NK production. So similar to CAR-T development, the basic of CAR-NK manufacture process includes NK cell extraction, CAR structure design and validation, transfection of the CAR molecule into NK cells, CAR-NK cells expansion and injection back to the patient after quality control. For every step in this process could be a challenging part and determine whether a CAR-NK product can be successful. And I would address three of them today, which could be the most important factors during the whole process. They are selection of antigen and antigen binding domain, cytokines, and CAR detection. The first and the most important thing in the CAR-NK production is the selection of the antigen. So the selection of an appropriate antigen is the crucial step in the development. And an ideal antigen requires high specificity, which means the target antigen should be expressed exclusively or predominantly on the surface of cancer cell and not on normal tissues to minimize the off-target toxicities. The target antigen should also not be essential for normal cellular functions to avoid potential toxicity to normal tissues. So here in this table, there are some typical target antigens which has high expression on tumor cells. And you can also see some of them are the successful drug target for CAR T cell therapy, like CD19. Also, after the selection of the appropriate antigen, how to develop the corresponding scFv is also very important. It also requires high specificity and affinity to ensure the efficient recognition of cancer cells. To this end, the binding affinity needs to be tested before the scFv can be constructed into the CAR molecule. Here shows an example of CAR T development of EGFR variant 3. The researchers use SPR to determine the affinity between scFv and EGFR and also their target EGFR variant 3. SPR result shows that the titratable binding of their product JCT02 scFv to the recombinant EGFR variant 3 compared with the negative GFP control and also shows no binding to the EGFR wild type. It turns out that the scFv only binds to EGFR variant 3 with good affinity, but not EGFR. This validation is very helpful in CAR development. So as we expected, after infusion of this product to the mice, the tumor size was decreased a lot. The next important factor during the production is cytokines. Since the NK cell represent only a minor fraction of the peripheral blood, so the generation of sufficient numbers of NK cells remains a major challenge for this kind of adoptive immunotherapy in humans. Currently, despite the various methods available to promote NK cell growth, the most widely used approaches is cytokine mixtures. So on the left side, we can see that the researchers have ever investigated on the function of IL-2 and IL-12 during the proliferation of NK92 cell line. The result shows that after adding IL-2 to the cell culture, the NK cell expansion got promoted in large scale, and also the IL-12 also have some effect. Besides promoting cell proliferation, cytokines can also increase the NK cell cytotoxicities by promoting the activation. On the right side shows an example. The researchers add a mixture of cytokines including IL-15, 18, and 27 to stimulate the NK cell. The expression level of perforin and granzyme B is largely elevated after the use of cytokines. This shows the cytokines can activate the NK cell. The next very important part during CAR-NK production is CAR detection. This is the step to assess the expression of CAR construct, which is very important in quality control. CAR expression levels and the consistency in CAR-NK cell need to be carefully monitored to ensure that the cells will function as intended in the clinical setting. Detection of CAR expression provides an important quality control measure to ensure that CAR-NK cells meet the required specifications before they can use for treatment. In addition, detection of CAR expression on NK cells after infusion is also needed. This can provide information about the persistence and distribution of the cells in the body. This information can help in monitoring the therapeutic efficacy of CAR-NK cell therapy. So here shows several detection methods to do the CAR detection. The first and widely used one is flow cytometry. So the mechanism here is use the fluorescently labeled antibody to recognize the parts of the CAR complex on the memory. And there are also some other methods like Western blot, quantitative PCR, and confocal microscopy. But some of them cannot do the single cell level. They can only monitor the CAR expression in a cell population. So next, let's see what we can do to facilitate the CAR-NK production. Biological is dedicated to providing one-stop research support, and now we have covered almost all the reagents and the services needed during the CAR-NK production, through CAR development to CAR-NK cell quality control. As we know, during the CAR-NK production, there are several key factors would determine the success of CAR-NK product. We are providing products and services to help our customers to overcome the challenge and get the best result. To facilitate the discovery of SCFV, we have 4 platforms, including phage display, beacon B cell, FAX B cell, and hybridoma platform. Since the SCFV sequences of most CAR-NK cells are derived from mice and have certain immunogenicity, SCFV needs to be humanized. Biological also provides high-quality monoclonal antibody humanization services that enable a high degree of successful humanization. As we showed before, a crucial determinant of CAR function is the affinity of the SCFV to its target antigen. We also provide SPR-BLI assay, which can effectively screen appropriate SCFV molecules. To facilitate the NK cell expansion, Biological also provides GMP-grade cytokines. Since cytokines need to be added into the cell culture and the cell product will be infused back to the patient, the quality of cytokines should be strictly controlled. Now we have 4 systems, including GMP-grade quality management system, quality control system, quality assurance management system, and product safety testing system to make sure we have the high quality, purity, and safety cytokines. To facilitate the CAR detection, we also have almost all the CAR-NK related target protein with high purity and labeled with built-in, which is ready to use to detect the CAR molecules on NK cell. Besides the variety of the target proteins, we also provide a customized service to develop anti-idiotype antibody, which can recognize the CAR molecule. So I will wrap up this webinar with an overview of Biological. Biological is an internationally recognized reagent supplier and contract research organization with focuses on recombinant protein and antibody services. As of now, we carry a large number of catalog products, including over 6,500 proteins, 14,000 antibodies, 47,000 genes, and over 600 ELISA kits. Up to September 2022, our products have been cited in over 10,000 publications, and some of these include high-impact journals such as Nature, Cell, and Science. Also, we are an ISO certified company supplying reagents to researchers in over 90 countries worldwide. We will keep working on providing high quality products and services to facilitate research and manufacture. Thank you for your attention.